Okay, welcome back. So last class we have discussed the process of fertilization, right? We have seen the fusion of male and female gamete, and we have seen double fertilization, we have seen triple fusion. So we have discussed all those. So next in this class we will discuss the process of formation of dicot embryo from zygote. Okay, the formation of dicot embryo. So in this video lecture we will discuss the formation of dicot embryo. Okay, so dicot embryo formation we will see. So embryo is formed from the zygote. Okay, all these things happen inside the embryo sac. Okay, so here we have zygote, which is the diploid cell. So we have zygote. Now zygote, what happened? First, the zygote will undergo mitotic division. Whatever the division that takes place in the formation of dicot embryo, all are mitosis division. Okay, there is no meiosis division. Whatever the division that takes place in the formation of embryo, that is only mitotic division takes place. The first, the zygote undergo mitotic division and form two cells attached together. Okay, the zygote will undergo mitotic division and form two cells. Make it round. Okay, make it round. And form two cells attached together. The upper cell known as terminal cell and the lower cell known as basal cell. Alright, the first thing for the happen, the zygote undergoes mitotic division and form the upper terminal cell and the lower basal cell. Good. Right? So next what happen? Again it will undergo further division. Mitosis only, okay? Only the mitotic division takes place in the formation of dicot embryo. So again those two cells will undergo further division. That division is known as, the name has been given, known as transverse, VERAC, transverse division. Transverse division. It is mitotic division only. The name is known as transverse division. Okay. So those two cells will undergo transverse division. Okay. The lower cell, first what happened? The basal cell, the one cell first it will form two cell. From one cell it will form two cell, and that two cell will give rise into again four cell, and so on. It will give rise into a long cell. Those cells are known as suspensor cell. Okay. So the basal cell, the first division we are taking place is the lower cell known as basal cell. So basal cell, after undergoing transverse division, it will form a long group of cell known as suspensor cell. One basal cell will give rise into two, then two cell again give rise into four, and the four again give rise into eight, and so on. Okay. And towards at the last point of the cell, towards the end point of the suspensor cell, we have a bigger cell as compared to the suspensor cell. Towards the end point of it, we have extra big cell known as haustoria cell. Okay, these are the end point of the cell known as haustoria cell. So these are the division of basal cell. Okay. Now the terminal cell, what happened? The terminal cell, after transverse division, which is a mitosis division only, mitotic division only. So after undergoing transverse division, what happened? It will form a four group of cell. It will form a four cell attached together. A mass of 4 cells attached together towards the upper region. Okay, a group of 4 cells attached together. So for our convenience, we are drawing in such a way so that we can understand. The actual diagram, it doesn't look like this, but it is for us to understand. Okay. So the terminal cell, after transfer division, it will form a group of 4 cells attached together. 1, 2, 3, and 4. Okay. That structure is known as, I'm writing here, pro-embryonal quadrant, pro-embryonal quadrant, that means 4 cell. Okay, understood? What happened? Zygote undergo mitotic division and form a 2 cell attached together. The upper cell is known as terminal cell and the lower cell is known as basal cell. Those 2 cells again undergo mitotic division known as transfer division, and the basal cell will form a long group of cells known as suspensor cell, and towards the end point of the suspensor cell known as haustoria cell are produced. Okay, and the terminal cell, after transfer division, it will form a 4 group of cells attached together known as pro-embryonal quadrant. Okay, again it will undergo further division, transfer division, which is mitotic division. Only the name is given as transfer division. Okay, it will undergo transfer division further. I am drawing below, and it will undergo transfer division further. First we will see the suspensor cell. What happened? The upper cell, which is in contact with the pro-embryonal quadrant, this cell, the upper cell, which is in the contact with pro-embryonal quadrant, it will become big. I hope we can able to see as compared to the suspensor cell. Only the upper cell which is in the contact, this cell, I am talking about this cell, which is in the contact with pro-embryonal quadrant, is become big, and the rest of the cell remains same. And the rest of the cell remain same. Okay, I hope you can see. So this was our suspensor cell. This is our haustoria. The cell which become big, the upper cell which in the contact with pro-embryonal quadrant, that cell is known as hypophysis cell. Okay, hypophysis cell. So this is the lower region of the cell, that is our basal cell. Now the upper part, pro-embryonal quadrant, after transfer division, it will again further divide into 8 cells. Okay, it will further divide into 8 cells. After 4 cells, now it will divide into, it will produce a group of 8 cells. Make it round. Okay, try to make it round. 8 cells, a group of 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. So after further transfer division, the pro-embryonal quadrant will give rise into a group of 8 cells attached together. Now we will call that cell as pro-embryonal octane or octet. Octane, that means 8, right? So pro-embryonal octet. And the lower region, the uppermost cell which was in contact with the pro-embryonal quadrant, it developed into hypophysis. Then the rest of the cell is same, suspensor is there, and then haustoria. Towards the end point of the suspensor we have haustoria cell, right? Okay, next step, what happened? Now next division is known as periclinal division. It is mitotic division only, Only the mitotic division takes place, okay? So periclinal division will takes place. Now after transfer division, it will divide into periclinal division. The division of the name of the division is known as periclinal. Now periclinal division develops the outer layer and the inner layer of the cell. Okay, the periclinal division result in the formation of outer and the inner layer. Okay, let me draw this diagram so that we can draw it nicely. So after periclinal, so after this periclinal, let me draw this. Okay, so after this, what happened? The periclinal division will take place. Periclinal division. So after periclinal division, what happened? The hypophysis cell, it divides into a group of 8 cells attached together. The hypophysis which was there, the hypophysis cell which was there, it will result in the division of 8 cells attached together, a mass of 8 cells, okay? But the diagram that we are drawing is for our convenience, okay? So that we can understand, but the diagram doesn't look exactly like this. They are, okay, one cell at the upper part, lower cell at the behind of it, like that. They are a mass of cell, a group of cell attached together. But that is for our convenience, okay? Try to understand. So hypophysis, what happened after periclinal division? It will result in the formation of a group of 8 cells. 1, 2, 3, 4, 5, 6, 7, 8. The suspensor cell remains as it is. The suspensor cell remain as it is. And then we have Haustorius cell. It remain as it is, okay? Now what is the function of suspensor cell, by the way? This suspensor cell, what they do, suspensor cell and the Haustoria, they push the embryo towards the endosperm, okay? We will see later. They push the embryo towards the endosperm inside the embryo sac. I'll try to put that image of embryo sac. So whatever these changes are taking place, they are taking place inside the embryo sac, okay? Try to understand. So these are the lower cell that the division took place. So after periclinal division, the hypophysis cell divides into the 8 group of cell, and then suspensor and the Haustoria remain as it is. The upper cell, proembryonal octate, it will result in the formation of layer, okay? It result in the formation of a layer, okay? We have 8 cells here. 1, 2, 3, 4, 5, 6, 7, 8. Now after periclinal division, a layer is formed. A layer is formed. A layer is formed between those cells. A layer is formed between those cells. Okay? So after periclinal division, what happened? The proembryonal octate result in the formation of layer, outer and the inner layer cell. Then the lowermost region, the hypophysis, it result in the formation of a mass of 8 cells, a group of 8 cells, and a suspensor cell and a Haustoria remain as it is. I'm not leveling again and again, okay? So now next we need to know the fate of all those cells. We need to know the fate of all those cells. I need a space, so let me write here. So this is our periclinal division. The hypophysis cell, the upper those 2 cells, upper those 4 cells, upper those 4 cells, it helps in the formation of root cortex. It helps in the formation of root cortex. Hypophysis means what? Hypophysis, hypo means below, the structure that is present below, okay? Epi means above, okay? So those four cells, the upper four cell result helps in the formation of root cortex later. Then the lower those two cells help in the formation of root cap and root tip, okay? So we need to know its function, we need to know its fate of the cell. So upper those four cells help in the formation of root cortex, and the lower those four cells help in the formation of root cap and root tip. Now what happened to that pro-embryo? The outer layer are known as, the name are known as dermatogen. The outer layer of the cell, the outer layer of the pro-embryo, that structure is known as pro-embryo, okay? The outer layer of pro-embryo are known as dermatogen. Dermatogen give rise to epidermis. It gives rise to epidermis, which is the outer layer of the any cell, okay? The outer layer of the cell. So dermatogen give rise to epidermis. Now, the half part of the lower region of pro-embryo, starting from here, okay? This is the half and this is the other half. That is the middle region, okay? That is the middle region. This is the lower half. This is the upper half. The lower half region, the lower half region, it helps in the formation of the lower before that, okay, wait, the upper half region, I'll come to lower region, wait, the upper half region, it gives rise into epicotyle, and plumule. Okay, the upper half region, it gives rise into epicotyle and plumule. The lower half region, it gives rise into radicle. Okay, the upper half region of pro-embryo, it gives rise into epicotyle and plumule, and the lower half region, it gives rise into radicle. Now next, what happened? Remember, all those changes are taking place inside the embryo. Inside the embryo sac. So we have seen the structure, and then we have seen the fate of a cell. Now that structure, we need to know the shape of the cell. That structure is known as, the shape of that structure is known as globular shape. Globular shape. The shape of that structure is known as globular shape. Okay, next, what happened? Okay, I will draw here only. So I hope you have drawn all those diagrams. Okay, you understood. Now next, I will show in this diagram only, in this particular diagram only. So I'm going to rub all those names. So you know the fate of each cell and the role of all those cells. So next, what happened? The lower eighth cell, is slowly and gradually result in the formation of a band-like structure. Okay, it slowly and gradually result in the formation of a band-like structure, and the upper region, the half cell, starting from here, it result in the formation of something, a heart-like structure like this. a heart-like structure appears. Slowly and gradually, what happened? The upper proembryo, half of proembryo, result in the formation of this structure, a heart-like structure, and the lowermost structure it bends. Okay, the structure something look like heart-like structure. Slowly and gradually, the structure of the proembryo changes, and changes are taking place inside the embryo sac. Remember, now from the globular shape, the structure will be called as heart shape structure. Remember the consequences of the change structure of the embryo. Okay, the first is globular shape. Okay, and the next structure it changes into a heart shape structure, and a heart shape structure. Although cell it become a mass of cell slowly. Okay. The next changes, I am showing in a particular diagram only, but you draw a different diagram. Okay. Next changes, what happened? Those structures become so compact inside the embryo sac. It becomes so compact that then what happened? Those structures they bends down. They bends down like this. They bends down. The half part of the proembryo again they bends down towards the same position. Okay, they bends down towards the same position of the other half of the proembryo, and then all suspensor cell and posterior cell ruptures. Okay, they bends down. This from the heart shape structure, the structure now it looks like this. Something like this structure, and that structure now we will call it as a horseshoe shape. Horseshoe shape structure. Now first globular shape, the consequences remember, first globular, then heart shape, then horseshoe shape, and finally what happened? And finally it will give rise into a dicot embryo. Finally, so I'm drawing it that side. It will give rise into a dicot embryo. It has a middle region. It has a middle region known as embryonal axis. Embryonal axis. And since we are discussing dicot embryo, so we have first cotyledon, then another cotyledon. The first disc it bends in such a way that it becomes like this. The other part, okay, it become like this, and the middle part become embryonal axis. So we have two cotyledon. The middle region of the embryonal axis, the middle part is known as embryonal node. Okay, and the region which is at the upper part here is known as plumule, which give rise to shoot structure, and the lower tip region is known as radicle that give rise to a root structure. Okay, plumule and radicle, and the structure which is here towards the upper region below the plumule, that structure is known as epicotyle, and the structure which is towards the radicle is known as, at the lower region, known as hypocotyle. Epi means above, hypo means below. So finally, from horseshoe shape structure, it will give rise into a dicot embryo. So this is the overall structure of formation of dicot embryo. So first it become first it become globular shape, then from globular shape it become into heart shape. The structure become heart like structure, and then it become into horseshoe structure. The upper half bends down and the other half again bend down towards the same position, and they become so compact, and they finally result in the formation of dicot embryo. Dicot embryo has embryonal axis at the middle region, and then at the middle part is known as embryonal node. Okay, the upper part is known as plumule. Upper tip part is known as plumule, which give rise to a shoot system. The lower tip is known as radicle, which give rise to a root system. Then the upper part of the plumule is known as epicotyle, and the lower part of the radicle is known as hypocotyle. So these are the overall process of the formation of dicot embryo, right? Understood? So first thing what happened, the zygote undergo mitotic division and result in the formation of two cells. The upper cell is known as terminal cell, the lower cell is known as basal cell, right? Those two cells again undergo transverse division. Then the basal cell result in the formation of suspensor cell and then haustoria cell. The terminal cell result in the formation of proembryonal quadrant, where the cell divides into a group of 4 cells. Okay, then it undergo further division, transverse division, and it result in the lower cell, which is the first cell which is in the contact with the proembryo. It result in the formation of what cell? It result in the formation of hypophysis cell, and then suspensor cell and haustoria cell remains as it is. The upper cell, the proembryonal quadrant, result in the formation of a group of 8 cells. Now we call it that as a proembryonal octet. Right, then again it will further divide, and that division is known as periclinal division. After periclinal division, the proembryonal quadrant result in the formation of outer and the inner layer cells. The outer layer cell is known as dermatogen, which give rise to hypodermis, which form the upper layer of the embryo. Okay, then the upper half result in the formation of epicotyle and plumule. The lower half result in the formation of radicle or hypocotyle, we can say radicle or hypocotyle. Okay, then that is the upper region. Then the lower region, what happened? The hypophysis cell the hypophysis cell result in the formation of 8 group of cell. The upper half result in the formation of it helps in the formation of root cortex, and the lower half region it helps in the formation of root tip and root cap, and the rest of the structure remains as it is. Next, what happened? And that shape is known as globular cell. Next, what happened? The lowermost cell, the bends down, and the uppermost cell, the structure, it appears a heart-shaped structure. That's why the structure is called as heart-shaped structure. And again, it will further become compact. What happened? The upper half region of the proembryo, it bends down. The other half, again, it bends down at the same time towards the same position, and finally it results in a formation of dicot embryo, where dicot embryo, it has the middle axis known as embryonal axis, the middle part of the embryonal axis known as embryonal node, the tip part of the embryonal axis is known as plumule, which gives rise to a shoot system, the lower tip part of the embryonal axis is known as radicle, which gives rise to a root system. The upper tip of the plumule is known as epicotyle, the lower tip of the radicle is known as hypocotyle. So these are the overall structure of dicot embryo. Diagram, make it clear, beautiful, step by step. Okay, explanations done, so answer you write by yourself. First diagram, then answer. Okay, so we have completed the process of formation of dicot embryo. So after embryo formation, what happened? A seed will form. So next class we will see seed, and then we will see fruit in the next video lecture.